My name is Moisés Mallo. I am a principal investigator at Instituto Gulbenkian de Ciência, and what we do in the lab is trying to understand how we make a body during embryonic development. In particular, we are mostly focused on things like why we have ribs in the thorax and not in the neck, or the limbs are in the place where they are, or even why snakes don't even have limbs. The basic model that we use for our research is the mouse, because it's the one allowing the best way to do genetic studies. The current work in my laboratory is mostly focused on understanding why we have the legs in the place where we have it, and the interaction between the formation of the legs and the external genitals that is important for the global functioning of the animals, because this is very interconnected and actually is essential for the vertebrates to be able to thrive in many different ecosystems. Fundamental research is the basics of every new knowledge that can eventually be applied to solve problems of humanity. One of the cases that everybody knows are antibiotics. They were found during basic research, but by chance, and then the impact that it had in humanity is huge. And you cannot find something, or you cannot look for something if you don't know that exists. So, this is the main power of fundamental knowledge, because you can find something just by chance that you didn't know that exists. It is difficult to replace animals in fundamental research because the whole body is very complex and has interrelationships. Nothing works on its own, and these interactions are very hard to reproduce outside of the animal. For instance, hormones or the immune system, you cannot put it into the in vitro systems. The best way to communicate the need of use of animals is to inform properly of the advantages and the reasons why we do that. One of the best ways to do it is just to give examples of things that really impacted the person that you are talking to. In this way, they can really understand why this research in animals was beneficial for them. We also have to give information that we are always trying to reduce the use of animals when possible. The use of new technologies like organ-in-a-chip or organoids can help and complement the use of animals, and this is especially true for cases in which we cannot use the animal itself for this kind of research. The best example is the understanding some things that happen during the development of the humans, that obviously you cannot do this in human embryos, and these kind of new technologies can help reproducing something that is happening in an embryo outside of the embryo.